Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. Students, today we are going to cover our next topic, that is lipids. So we will try to discuss and study lipids in detail. So here is a detailed account for lipids from class 11 biology for federal boards, Pakistan. So here, lipids. Lipid is the collective name for variety of organic compounds such as fats, oils, waxes, and fat-like molecules such as steroids found in the body. So, what we studied? We studied that lipid is the collective name for variety of organic compounds. So lipids includes different types of organic compounds, a variety of organic compounds, and this variety of organic compounds includes examples are fats, oils, waxes, steroids, etc. Therefore, it is defined as a heterogeneous group of organic compounds which are insoluble in water but soluble in organic solvents such as acetone, alcohol, and ether, etc. So, what is the next point? The next point is because these contain a variety of organic compounds and lipids is the collective name for a variety of organic compounds. So, we say we can define lipids as a heterogeneous group of organic compounds. It is a heterogeneous group of organic compounds which are insoluble in water, and insoluble in water means they have no affinity for water molecules. So as they have no affinity for water molecules, so they are not dissolved in water, means they are insoluble in water, and thus they are regarded as hydrophobic compounds but soluble in organic solvents. However, these are soluble in organic solvents such as acetone, alcohol, and ether, etc. Lipids are composed of carbon, hydrogen, and oxygen as carbohydrates. So, lipids, like carbohydrates, are composed of carbon, hydrogen, and oxygen. So we studied in previous tutorials that carbohydrates are composed of carbon, hydrogen, and oxygen. So likewise, lipids are also composed of carbon, hydrogen, and oxygen. However, they have relatively less oxygen in proportion to carbon and hydrogen than do carbohydrates. However, they have relatively less oxygen in proportion to carbon and hydrogen as compared to carbohydrates. They have relatively less oxygen in proportion to carbon and hydrogen. For instance, trastyrene is a simple lipid which shows molecular formula as C57H110O6. So it is an example. We can see that trastyrene is a simple lipid and it shows molecular formula C57H110O6. So there is a very low proportion of oxygen. There are only 6 atoms of oxygen as compared to 57 atoms of carbon and 110 atoms of hydrogen. So oxygen are present in low proportion. They have only 6 atoms in trastyrene. Due to higher contents of carbon and hydrogen, they contain double amount of energy than carbohydrates. So when compared to carbohydrates, so for the same mass of carbohydrates, when they are compared with lipids, lipids contain double amount of energy. They contain high contents of carbon and hydrogen, and due to the presence of high contents of carbon and hydrogen, lipids contain double amount of energy as compared to the same amount of carbohydrates. In general, lipids are components of cell membranes, act as energy stores, steroid hormones, and are also involved in protection, waterproofing, insulation, and buoyancy. So, lipids perform different functions. These are components of cell membranes. Phospholipids and cholesterol are the lipids that are the components of cell membrane. So, phospholipids and cholesterol are the components of cell membranes. Triglycerides act as energy stores. There are steroid hormones, and lipids are also involved in protection, waterproofing, insulation, and buoyancy. Some common lipids are acyl glycerols, waxes, phospholipids, terpenes, prostaglandin, and steroids. So first we will have an account of acyl glycerols. The most abundant lipids in living things are acyl glycerol. Acyl glycerols are the most abundant lipids in living things. Chemically, acyl glycerols can be defined as esters of glycerol and fatty acids. Chemically, we define acyl glycerols as esters of glycerol and fatty acids. So here we can see uh, this is glycerol, trihydroxy alcohol. Okay, this is glycerol, and it is reacting with fatty acids. Well, so here in this example, we see there are three fatty acids. So three fatty acids are combining with one glycerol molecule, and a reaction takes place. Water is released, and triacyl glycerol is formed. So acyl glycerols are the esters of glycerol and fatty acids. And this reaction is called esterification. The bond between glycerol and fatty acids is called ester bond. Here we can also see this is an alcohol, and it is reacting with a carboxylic acid, methanol. Alcohol is methanol, and the carboxylic acid is ethanoic acid. So dehydration reaction occurs. Water is released along with the formation of methyl ethanoate and ester. An ester is the compound produced as the result of a chemical reaction of an alcohol with acid, and a water molecule is released. Such a reaction is called esterification. So, an ester is formed when alcohol and acid react with each other, one another, an ester is formed. So an ester is the compound produced as the result of a chemical reaction of an alcohol with acid, and a water molecule is released. Such a reaction is called esterification. Glycerol is a trihydroxy alcohol which contains three carbons, each bears a hydroxyl group. So glycerol is a trihydroxy alcohol. It is a trihydroxyl alcohol. There are three hydroxyls, one, two, three, trihydroxy alcohol, and each bears hydroxyl group. Hydroxyl group is present with each carbon atom in glycerol. A fatty acid is a type of organic acid containing one carboxylic acid group attached to a hydrocarbon. So what are fatty acids? Fatty acids are the types of organic acids that contain one carboxylic acid group attached to a hydrocarbon. So here we can see this is a carboxylic acid, means fatty acid. Long chain carboxylic acids are called fatty acids. Fatty acids contain even number of carbons from 2 to 30. Fatty acids contain 2 to 30 carbons, and they contain even number of carbon atoms. Each fatty acid is represented as RCOOH, where R is the hydrocarbon tail. This R is the hydrocarbon tail, hydrocarbon chain. When a glycerol molecule combines chemically with one fatty acid, a monoacyl glycerol or monoglyceride is formed. When two fatty acids combine with a glycerol, a diacylglycerol or a diglyceride is formed. And when three fatty acids combine with one glycerol molecule, a triacylglycerol or triglyceride is formed. So here, if only one fatty acid combines, monoacyl glycerol or monoglyceride. If these two combine, then diacylglycerol or diglycerides. And in this example, we see that three fatty acids are combining with glycerol, so a triacylglycerol is formed. Triacylglycerols are also called neutral lipids as all three hydroxyl groups of glycerol are occupied by fatty acids and no charge bearing hydroxyl group is left. So there are no charge bearing hydroxyl groups, and all three hydroxyl groups of glycerol are occupied by fatty acids. Here we can see all hydroxyl groups are occupied by fatty acids. So there are no charges, there are no charge groups, and this compound is called neutral lipid. These compounds are called neutral lipids. There are some properties and types of fatty acids we will study here. About 30 different fatty acids are found. About 30 different fatty acids are found. Fatty acids vary in length. Acetic acid and butyric acid are simplest fatty acids, whereas palmitic acid and stearic acid are most common fatty acids. So, acetic acid and butyric acid are simplest fatty acids, uh, and palmitic acid, which is a C16 fatty acid, and stearic acid, which is a C18 fatty acid, are most common fatty acids. Well, acetic acid is a 2 carbon, and butyric acid is a 4 carbon acid. Some properties of fatty acids are increased with an increase in number of carbon atoms, such as melting point, solubility in organic solvents, and hydrophobic nature. So there is a link between the number of carbon atoms in fatty acids and their properties. So some properties of fatty acids are increased with an increase in number of carbon atoms, such as melting point, solubility in organic solvent, and hydrophobic nature. Some common fatty acids are given in the table 2.6. Here we can see there are some fatty acids. These are the saturated, palmitic acid, stearic acid. These have carbon atoms fully saturated. Uh, they contain maximum number of hydrogen atoms chemically reacted with carbon atoms in these compounds, in these carboxylic acids, in these fatty acids, and there are carbon-carbon single bonds. All carbon-carbon bonds are single. While in unsaturated fatty acids, they have at least one single or triple bond in them. So examples of unsaturated fatty acids are oleic acid and linoleic acid, and these are present in vegetables, plant sources. These come from plant sources, olive oil or vegetable oils. And here we can see their melting points. If there are more carbon atoms, their melting points are high. For example, 16 carbon fatty acid contains has a melting point 63, while stearic acid that contains 18 carbon atoms has the melting point of 70. Here we see that unsaturated fatty acids relatively have low melting points. They have low melting points due to their unsaturated nature. Fatty acids are either saturated or unsaturated fatty acids in which all of the internal carbon atoms possess hydrogen side groups are said to be saturated fatty acids. So they contain hydrogen side groups. All of the internal carbon atoms contain hydrogen groups because they contain the maximum number of hydrogen atoms that are possible. For example, palmitic acid. So they contain maximum number of hydrogen atoms. Saturated fatty acids tend to be solid at room temperature and are more common in animal lipids. Then we see unsaturated fatty acids have one or more pairs of carbon atoms joined by a double bond. They have one or more pairs of carbon atoms, and these are joined by double bonds. They therefore are not fully saturated with hydrogen. They are not fully saturated with hydrogen means that they do not contain the maximum number of hydrogen atoms due to the presence of double or triple bonds. And example includes here oleic acid. Unsaturated fatty acids are liquid at room temperature and are more common in plant lipids. They are a liquid at room temperature and are more common in plant lipids. For example, oils, triglycerides containing hydrocarbon chains melt at a low temperature. Triglycerides that containing hydrocarbon chains melt at a low temperature. This is useful for living things. So it was an account for acyl glycerols or triglycerides. Now we see waxes. Waxes are highly hydrophobic compounds. Waxes are highly hydrophobic compounds. There are two types of waxes. Natural waxes are simple lipids. They are typically esters of long chain fatty acids and long chain alcohols, such as beeswax found in honeycomb and cutin on leaf surfaces of plants. Well, waxes are of two types: natural waxes, synthetic waxes. Natural waxes are simple lipids. They are typically esters of long chain fatty acids and long chain alcohols. And examples include beeswax found in honeycomb and cutin on leaf surfaces of plants. Cutin is present on leaf surfaces of plants. These are chemically inert and resistant to atmospheric oxidation. Waxes have protective functions in plants and animals. Synthetic waxes are generally derived from petroleum or polyethylene, and its example is paraffin wax, which is used to make candles. Next group of lipids is phospholipids. Phospholipids are derived from phosphatidic acid. A phospholipid is formed when phosphatidic acid combines with one of the four organic compounds such as choline, ethanol, amine, inositol, and serine. So choline is a nitrogenous base. It may combine with phosphatidic acid, ethanolamine, and amino alcohol, inositol, and amino alcohol, and serine is an amino acid. So, when phospholipid is formed when phosphatidic acid combines with one of the four organic compounds such as choline, ethanolamine, inositol, or serine. A phosphatidic acid molecule is most similar to diglyceride, that it contains a glycerol, two fatty acids esterified with first and second hydroxyl group of glycerol, and a phosphate group esterified with third hydroxyl group of glycerol. So, what is a phosphatidic acid? Phosphatidic acid is most similar to diglyceride. Here we can see this is a glycerol, and there are two fatty acids at carbon number 1 and 2 of glycerol, while the third carbon in glycerol is chemically linked with choline. Okay, so this is known as phosphatidylcholine. It is also known as lecithin. So third of carbon at carbon number 3, choline reacts with the hydroxyl groups of glycerol, and phosphate group is esterified with third hydroxyl group of glycerol. Most common types of phospholipid is phosphatidylcholine. Here we can see this is a phosphatidylcholine, also called lecithin, in which choline is attached to phosphate group of phosphatic acid. One end of the phospholipid molecule containing the phosphate group and additional compound is hydrophilic. So here we see that one end of the phospholipid molecule containing the phosphate group, this end is hydrophilic, polar, and readily soluble in water. The other end containing the fatty acids, this end that contains the fatty acids, side chains, is hydrophobic, non-polar, and insoluble in water. These phospholipids are major constituents of lipid bilayer of cell membranes. So we already studied that phospholipids and cholesterol are present in cell membranes. So these phospholipids are major constituents of lipid bilayer of membranes. Next is terpenes. All the terpenes are synthesized from a 5 carbon building block known as isoprene unit. This unit condenses in different ways to form many compounds. So all the terpenes are synthesized from a five carbon building block known as isoprene unit, and this unit condenses in different ways to form many compounds. Two isoprene units for a monoterpene, for example, menthol, four form a diterpene, and examples include vitamin A, phytol, or chlorophyll, and six form a triterpene. Amprene, natural rubber is a polyterpene, and this is isoprene unit that contains uh, five carbons. So it is a five carbon building block. Next comes steroids. Steroids are lipids of high molecular weight which can be crystalline. A steroid nucleus consists of 17 carbon atoms arranged in four attached rings. Three of the rings contain six carbon atoms, and the fourth contains five. So steroids are lipids of high molecular weight, and these can be crystalline, and which can be crystalline. A steroid nucleus consists of 17 carbon atoms, and these are arranged in four attached rings. Of these four attached rings, three of the rings contain six carbon atoms, and the fourth contains five carbon atoms. The length and structure of the side chains that extend from these rings distinguish one steroid from other steroids. So, the length and structure of the side chains determines distinguish the type of steroid. It distinguishes one steroid from other steroids. These structures are synthesized from isoprene units. And cholesterol is a structural component of cell membrane. Cholesterol is also a structural component of cell membrane. We studied phospholipids are present in cell membrane. So cholesterol is also a structural component of cell membrane. Cholesterol is the precursor of a large number of equally important steroids, which include the bile acids, male sex hormone, testosterone, female sex hormone, progesterone, and estrogen. So these are the precursor of a large number of equally important steroids and other compounds. Hormones, bile salts, which emulsify fats, and vitamin D, which helps to regulate calcium metabolism, are also steroids. So bile salts are steroids, and vitamin D is also a steroid. And bile salts emulsify fats, and vitamin D helps to regulate calcium metabolism, and these are steroids. And in the last, we will study prostaglandins. Prostaglandins exist in virtually every mammalian tissue, acting as local hormones. So prostaglandins act as local hormones, and they exist in virtually every mammalian tissue. Prostaglandins are derived from arachidonic acid. So these are derived from arachidonic acid. Their functions vary widely depending on the tissue. So their functions depend on the tissue. Depending on the tissue, their functions vary. Some reduce blood pressure, whereas others raise it. In the immune system, various prostaglandins help to induce fever and inflammation, and also intensify the sensation of pain. So, in some cells, these reduce blood pressure, whereas others raise it. In the immune system, these induce fever and inflammation, and these also intensify the sensation of pain. They also help to regulate the aggregation of platelets, an early step in the formation of blood clots. They also help to regulate the aggregation of platelets, and aggregation of platelets means it is an early step in the formation of blood clots. In fact, the ability of aspirin to reduce fever and decrease pain depends on the inhibition of prostaglandin synthesis. There is also a short note. Prostaglandins play a pivotal role in inflammation, a process characterized by redness, heat, pain, and swelling. The changes associated with inflammation are due to dilation of local blood vessels that permits increased blood flow to the affected area. The blood vessels also become more permeable, leading to the escape of white blood cells from the blood into the inflamed tissue. Aspirin is anti-inflammatory, analgesic, and antipyretic. Aspirin inhibits prostaglandin synthase salicylate. so it inhibits the enzyme. This drug affects the metabolism of arachidonate via the lipoxygenase pathway by inhibiting the conversion of 12 hydroxy to 12 hydroxy 5 8 10 14 icosa tetranoic acid. So it was just just an additional information today. Again, prepare it, study it, and if you have any questions, you can go for your questions, and we will try to provide you the answers and solutions. Thank you, Allah Hafiz.